Good afternoon, everyone, and thank you for joining us today. I'm Pat Furlong from the PPMD team, and we're thrilled to be able to present to you from Capricor Therapeutics interim analysis today. With us, we have Linda Marban, President and CEO of Capricor Therapeutics, who's going to start off discussing these interim results on the HOPE-2 study. So with that, I'm going to turn it over to you, Linda, and we'll take questions along the way as well as at the end.
Thank you so much, Pat, and thank you, PPMD, for giving me this opportunity to present the interim analysis data and also for the support, the energy, and the ability to move forward the program within the Duchenne muscular dystrophy using CAP-1002. We're a publicly traded company. Our symbol is CAPR, and this is a standard forward-looking statement. Capricor was founded a little bit more than a decade ago with the idea that we would use stem cell-based therapies and exosome-based therapies to treat patients with immune, inflammatory rare disease, and we are now focusing hard on Duchenne muscular dystrophy. Today we're going to be talking about CAP-1002, the product in our pipeline which has been in patients with DMD for several years now, first in the HOPE-Duchenne clinical trial and now in HOPE-2. We have another product in our pipeline, the exosomes. We won't be talking about those today, but we're very excited about those and have a record of the last several years of publishing in the exosomes and also their potential efficacy in DMD. So we look forward to bringing those to this community within the next few years as well. So let me tell you a little bit about CAP-1002 and our DMD program. Feel free to interrupt with questions if that's appropriate or type them up and I'll get them at the end. So HOPE-2 clinical trial is designed as a randomized, double-blind clinical trial to determine whether CAP-1002, 150 million cells delivered intravenously or placebo, are delivered every 3 months. And we are looking for measures of safety, of course, and focusing on efficacy. The patients in HOPE-2 tend to be older patients, tend to be non-ambulant. I'll be talking about that in a few moments. So therefore, the primary efficacy measure is something called the performance of the upper limb, which is a measure of skeletal muscle function in those that are no longer to ambulate well or at all on their own. Hold on. The mechanism of action of CAP-1002 is that it is a strong immunomodulatory agent. That means that it helps to control bad inflammation and encourage good inflammation, which encourages muscle repair. We know that the mechanism of action of CAP-1002 is likely to be from the secreted exosome, again, the pipeline product I mentioned a little bit ago. And exosomes contain non-coding RNAs, proteins, and other signaling molecules that can change the behavior of cells. Our cells do not work by stemness. That means they do not become part of an injured tissue and grow and divide. What they do is that they drive the cells to go back into the cell cycle that helps them make new muscles, and also we believe are strongly anti-inflammatory and immunomodulatory, as I mentioned a little bit ago. CAP-1002 has been in several clinical trials, starting the world and adult heart disease, and now in 2 Duchenne muscular dystrophy trials, and more than 150 subjects have been treated with the cells with a very strong safety profile. Well, let me just remind you, for those of you that are sort of just now tuning in to the opportunity for CAP-1002 and DMD, that we started off doing a clinical trial called HOPE-Duchenne. Now, Capricor founded itself on the general concept that the cells that we have, which are made from heart tissue, would primarily be treating heart disease. So we developed a paradigm along the way where we would inject the cells into the coronary arteries of the heart so that we could get the cells as close as possible to the site of injury and potentially address the disease of the cardiac tissue. What we found in preclinical analysis, which I don't have here, but you can definitely find on our website or through questions, or Pat also has access to a lot of our preclinical data, was that we had beneficial effects in the MDX mouse, both in the heart and in skeletal muscle. So that led us to design the first trial, HOPE-Duchenne. It was an open-label trial of 25 boys. They had to have a certain level of heart disease, so they were older by the luck of cardiac dysfunction, which is taken place generally in the later stage of the disease. They had to have been on stable steroids. They needed to have scar on their hearts and still a healthy enough ejection fraction that they weren't at risk of full-fledged heart failure. They were then randomized, either received 75 million cells into their coronary vessels. That means they went into a cath lab and we put a catheter up into their heart, not we, but the physicians that are qualified to do so. And 25 million cells were put down each of their 3 coronary arteries for a total dose of 75 million cells. This was done one time. The other half of the patients were randomized to usual care. That means we just watched them. They didn't get a placebo injection because we didn't want to put them through the risk of going into the cath lab. And then we looked for primarily safety, of course. We had never treated patients with Duchenne, and they had never actually undergone any kind of therapeutic with coronary catheterization. So we focused hard on safety, but we built in a variety of exploratory efficacy measurements, focusing first on heart using MRI, the performance of the upper limb. We did the 6-minute walk test because in those days we didn't know that most of our patients would be non-ambulant. And we also looked at pulmonary function tests, quality of life, and biomarkers. And what we found was very interesting. We had an older patient population. They were about 18 years of age. Most of them in the treated group were using wheelchairs most of the time. And in the usual care group, they tended to be a little less impacted. This was just one of those luck of throwing the dice and where everybody ended up. They all had a pretty similar amounts of cardiac scar and ejection fraction. And what we found from HOPE-1, and again, I don't want to take the time today to spend a lot of time on the data from HOPE-1, but I wanted you to see, where the general concept of HOPE-2 came from, is that we saw early indications of improvement in cardiac scar and systolic wall thickening, and remember that because you're going to see it again in a little bit. Skeletal muscle function improved in the performance of the upper limb. And the cells were generally safe and well tolerated, although we didn't love some of the side effects that seemed to go along with the intracoronary infusion procedure. So we really wanted to see the program move forward, but really take a step back from the intracoronary injection. And we also did one injection, as I mentioned to you, and so we saw the effect kind of peak around 3 months and then begin to drop off, and so we knew we needed to do a repeat injection paradigm. So that took us back into the lab, and we did the work that led us to HOPE-2, which I'm now going to be sharing the results of you with. And just to recapitulate, HOPE-2 is a randomized, double-blind, placebo-controlled trial. It's intravenous injection. Because we're going IV, we are not worrying about causing any potential damage to the heart, so we're able to double the dose of cells. So in HOPE-1, we gave 75 million cells, and in HOPE-2, we're giving 150 million cells. And now I'm going to share with you what we have found. And the patients are dosed 4 times, although I'll remind you that the data that you're going to see today is the 6-month data, which is after infusion with 2 doses of cells, and then looking at the data after that. So we had 9 sites in HOPE-2 that were active. Dr. McDonald, our national PI, was the most active with 8 subjects enrolled, but we had patients enrolled and treated at other sites. So that's a very good thing from a regulatory perspective because it shows that wherever we deliver these cells, we seem to be seeing the same type of results. And what I can tell you that the cohesiveness of the data that you're going to see means that wherever the cells are delivered, they seem to be doing the same thing. So there's no magic going on at any one of the sites. We had 26 subjects that were enrolled before we were able to do this interim analysis. That means that 6 screen failed and 20 were treated. The screen fails weren't anything specific. I would say if we had to sort of lump it into one corner or another, it would be a pro-level entry score, but we also had a few other things, so nothing coming out specifically for screen failing. And as I mentioned, we had 8... Go ahead.
Would you just give a little more detail on the screen fails? And you mentioned entry score. Can you just sort of explain that a bit so everyone's on the same page going forward?
Absolutely. So I'm a little nervous to try and go off my computer to pull up the reason for the screen fails, but I will when we take a break for questions. What I will say is that but I can talk about the inclusion criteria and the entry-level pull score. So the entry-level pull scores are a low of 1. That means you pretty much have lost upper limb function in the mid-level. It means you might still have distal, but your ability to lift and move your arms independently is pretty attenuated. And a 6 means, on a scale of 1 to 6, that you haven't lost any of it, that you're absolutely still fully able to move your arm without impact of the disease. We shaved off both ends, so we didn't take in those patients that had a score of 1 because we didn't think that we could probably do much better at that point. We were concerned that they might be too far along to be able to really measure an improvement. And then conversely, on the flip side, if they had full functioning, there was no delta to improve. So we took full entry scores from 2 to 5. And I, again, will get the other reasons for the screen fails. So I know 2 of the patients screened out because of full entry score, and there were a couple other reasons. One of them was like somebody had a respiratory infection or something like that, couldn't do it and was going to potentially come back. But before we hang up, I promise I will switch over and find it. I just don't want to lose the slide.
No, thank you. I think that's really helpful to understand based on the pool score, right? With very high functioning, you're not going to be able to measure improvement, and very loss of considerable function as a very low pool score, you're really unable to detect change. So I think that's very helpful. Thank you.
Yes, absolutely. So we ended up having 8 patients that were treated with cells and 12 that got placebo. That's just the luck of the randomization draw, but it gave us a really good indication of what was happening moving forward. And in addition, what's nice about that is because there's so few placebo-controlled trials in the Duchenne muscular dystrophy space, we really are grateful to have that bank of data to be able to look back and benchmark against natural history. So that was very, very good luck for us, I would say. So let me walk through where we are and what we have today. So the intention to treat population were 20 subjects. That was the 26 minus the 6 that screened out. We're able to follow all 20 for safety. A lot of the data that I'm going to be showing you today is on something called the PP, or per protocol population. Those are 17 of the 20 subjects. As many of you know, and we'll talk about a little bit later, we had some safety issues that we needed to address. The good news is with the hypersensitivity allergic reaction that happened in one patient in the HOPE-2 trial and one patient in an open-label extension of the HOPE-Duchenne trial, we were able to initiate a pre-medication regimen, which has been very successful. But while we were down trying to figure all that out, 3 patients missed scheduled infusions, and so we didn't include them in this analysis population. What I can tell you, because I know you're going to ask, is that it didn't change the data very much at all. In fact, the ITT data, the intention to treat population data, was almost exactly the same as the PP data. I just wanted to present the cleanest data set we had. And the safety population data is available for interim analysis by visit. So on day 1, that means 12 patients were infused with placebo, although they don't and will continue to not know who they are. And 8 patients got cells, and the same thing, they don't and will continue to not know who they are for 20 patients. So we have 20 patients data total. We have 17 patients now where we have data 3 months later, so after 1 dose, you can see what happened. And then for 13 patients, we have data after 2 doses, and that's where we focus the interim analysis. Now, why did we do the interim analysis? We did the interim analysis because, frankly, we wanted to make sure before we had patients come in and get infused and families put their hopes on the line that we actually were seeing a potential therapeutic benefit. There was a fair bit of questions around the HOPE-Duchenne data because of the performance of the upper limb improvement with a cell that was designed potentially to treat cardiac symptomatology. It was wondered if the volitional aspects were utilized, and therefore patients perhaps were not representing their standard curve of ability, but rather an accelerated. So this time we were able to rule that out. We wanted to look at 6 months. We didn't want to wait till more patients got to a year because we wanted to be able to stop if it wasn't going to be successful. So let me talk to you a little bit about the patients. This is where the clinical trial gods were being very kind because we ended up with a really lovely homogeneous group of patients. So we feel like the data that we have is in fact data that is representative of the patient population we're looking to treat. The kids in this clinical study were about 14 years old on average, and they didn't vary very much at all between the treated and the placebo. They all had BMIs around 22.6. It's one of the side effects of steroids, and so we expected them to have fairly high BMIs. And as I mentioned, we have pull entry scores between 2 and 5, but in this situation, we've stratified them. So we didn't want to end up having all of our patients at one end or another of that spectrum. So patients were stratified in the randomization criteria of either a 2 to a 3 or a 4 to a 5. As I mentioned, all of the patients were on stable steroids. The reason we do that is because we don't want to be assailed in any way as to not maintaining standard of care. We want all the patients to get the best that medicine has to offer them. And so therefore this was a requirement, and the FDA likes to see that, by the way. The regulatory agencies like to see it because they recognize that most patients are on steroids. 80% of the patients were non-ambulant. It's interesting because it's pretty much the same as it was in HOPE-2, so we think that that's very interesting. And these are the patients that we're targeting with our first label. We're looking for these later stage non-ambulant patients with a significant amount of muscle fibrosis. We couldn't take patients with spinal surgery or symptomatic heart failure for reasons of measurement and also for reasons of being able to ascertain cardiac structure and function. So I'm going to be showing you a fair bit of data, and because in the interest of time, there's a lot of you, so I want to make sure I leave time for questions. I'm sort of giving you the laundry list of measures that we looked at here for efficacy. So the primary efficacy endpoint originally of HOPE-2 was the performance of the upper limb version 1.2. We also measured the performance of the upper limb 2.0. I make the analogy, which I think everybody understands, that you always want the iPhone 8 instead of the iPhone 6, so the pull is much the same. Pull 1.2 was the early version, pull 2.0 is sort of the better, more improved version, and in fact that data bears out here. So it's been a very successful modification of a very good scale anyway.
And you can also comment, Linda, on your RMAT meeting at the FDA that the FDA did recommend 2.0 pull as opposed to 1.2. So this is also a regulatory recommendation.
Right, exactly, Pat. So as usual, we're channeling each other because I was just going to start talking about the FDA meeting. So we met with the FDA in December. We have something called RMAT, Regenerative Medicine Advanced Therapy designation, that is part of the 21st Century Cures Act. If you're not familiar with that, it's like breakthrough therapy for cells and genes, but it's even better than breakthrough because it really is a very narrow funnel. You have to have a cell or a gene therapy, and there's very specific rules that they have to get this designation, and then they help you get your therapies faster to approval. They told us a bunch of very important things, and this is a great point to emphasize them. They asked for us to switch the primary efficacy endpoint from pull 1.2 to 2.0. They liked it better because of the reduction in variability and the more specific tasks assigned to each muscle group. They wanted other measures of skeletal muscle strength. They didn't just want us to use the pull. They wanted to be able to see if there was a correlation or an analogy between what we would see in pull and what we would see in other skeletal muscle functional measures. So for that, we measured absolute grip strength, tip-to-tip pinch strength, and some other ones like elbow flexion and things like that. They asked us to, and of course we were interested in measuring pulmonary function. We have 3 measures in the interim: peak expiratory flow, % predicted, inspiratory flow reserve, % predicted, and forced vital capacity, % predicted. What's interesting is I went back through our FDA meeting minutes in preparation for this and some of the other meetings I've been doing the last few days, and they specifically called out PEF as one of the ones that they like as a secondary endpoint. And then we measured cardiac structure and function. This was for the purpose of seeing if we can make inroads into treating the heart disease, and also because, of course, this would be the validation of what we saw in our HOPE-2 clinical trial. So all of these things were measured, and then, of course, quality of life assessments, which are still under review. I don't have that data today. It's a lot of data to review. So let me, tell you the most exciting part first, and that is that we have already, with very few patients, but already statistical significance at month 3, so after 1 dose, and continuing to month 6 with 2 doses of the cells when compared to placebo. Remember, these are very small sample sizes, and I'll give you a little bit of the same heads up that our statistician gave me before I got this data, which is that typically in an interim analysis, the only thing you can do is determine whether or not the data is good enough to proceed. And so you're not looking for statistical significance at this point. You're looking for the basic slightest trend. You know, if you happen to be a hiker or something, you see a mark on a tree, you can go down that pathway. That's kind of what you look for in an interim analysis. So the fact that we see statistical significance here speaks to the quality and the consistency of the data that's been measured and gives us a real guidepost that this trial and this effort to treat Duchenne should continue with CAP-1002. Now let me also take you forward and talk to you about some of the other data that we saw. So again, we wanted to see if there was any correlation in other measures of skeletal muscle strength. And again, we saw some trending and even a little bit of statistical significance in grip strength and tip-to-tip pinch strength, both significant or nearly significant at the 6-month time frame. This is very interesting and important because this is the first time, in fact, in any clinical trial with CAP-1002 that we've done more than 1 dose. And so it's really gratifying to see that the effect not only continues, but appears to be accelerated or augmented by repeat dosing. So we're very much looking forward to seeing more 9 and 12 month data to determine if these trends can actually continue. But now we have really a war chest of skeletal muscle indicators that our patients are doing better than the placebo-treated patients. And they're all our patients. I think they are patients. The treated patients are doing better than the placebo-treated patients. Every patient in this trial is our patient. So in terms of respiratory function, again, trying to set this up, this data is far better in terms of showing some hints of efficacy than I was ever hoping to see. As you know, and all of you are very well aware of this, measuring diaphragmatic function in patients with Duchenne is very hard. These tests are hard for the patients, for the boys to do. Sometimes they don't like to do it. It requires breathing very hard. It requires specially trained technicians that know how to measure it. So I think everybody knows that while this is perhaps number one, what one could argue whether respiratory or cardiac function are the ones that you worry about the most long term, certainly they are 1 and 2 in some order of that nature. It's very hard to measure, and so we were very gratified to see that we were seeing trends in peak expiratory flow, and then, as you'll see in this next slide, we also see changes in inspiratory flow reserve. So inspiratory flow reserve is basically how well you're breathing in, how your diaphragm moves, and PEF, the one you just saw, is how well you breathe out or expire, or exhale is probably a better word there. So real measures of perhaps certainly trending and looking like improvement in diaphragmatic strength, which is very exciting. We are still trying to analyze the forced vital capacity. I didn't put the graph up because it's confusing. You have to do very specific analyses of height that's based on percent predicted. And so we have Dr. Hank Meyer at Children's Hospital of Philadelphia doing actual individual spirometry reviews right now to see if we can tease out what we're actually seeing in forced vital capacity. The initial read of the data, the forced vital capacity, was not as good as the inspiratory flow or the peak expiratory flow, and that doesn't seem to match up. So I'm not showing the data because we don't understand it yet, but please stay tuned as that story becomes more clear. And finally, when we first did the HOPE-Duchenne clinical trial, we learned a lot about hearts of patients with Duchenne muscular dystrophy. Very few groups had done systematic analyses of the MRI from Duchenne patients. There's a lot of field experience on looking at heart attacks and heart failure, which is a very, very different picture. And when you think about doing an MRI of the heart, you can think about a donut. And that's basically what we're looking at. And we usually look for scar within that donut, and then we can quantify scar versus healthy and come up with a calculation of the amount of scar. Additionally, and we didn't do scar in this clinical trial because you have to use a dye called gadolinium, and gadolinium has some side effects that we just don't want to expose kids to. So we're not looking at scar. We showed changes in scar in HOPE-1, and we thought that was good enough. But what we learned is that the hearts of Duchenne patients, because of the dystrophin mutation and the aggregation of that scar, is very different than heart attacks. What we learned is that a very important measure of cardiac output is something called systolic wall thickening. So with MRI, we can actually look really closely, like laser focus on different parts of the heart. And basically, even though it's a donut, we can kind of divide it into 4 walls: anterior, lateral, inferior, and septal. And basically, the scar pattern in Duchenne, which should appear to be random, but is very interestingly not random, it is a progression of scar from anterior to lateral to inferior to septal. By the time you see inferior and septal, you have a lot of scar, and usually you have reduction and ejection fraction. But if you have scar in anterior and lateral, you still have pretty normal ejection fractions like our patients. What we see here is that we see improvement in function in the anterior and the lateral walls, very similar to what we saw in the HOPE-Duchenne clinical trial, which was very, very exciting and rewarding to know that we're being able to validate that data and seeing actual improvements in cardiac function. And finally, we saw an increase in myocardial mass, that is the LV mass, or basically increasing a little bit the size of the donut. Now, if you were showing me this data from adult heart failure patients, I would say, uh-oh, this is not a good thing, because those hearts, when they get bigger, it means that bad things are happening. It's pathologic enlargement. But in this situation, we believe, and our KOLs believe, our key opinion leaders, those cardiologists that we've shown this data to, that it's actually a good thing, because it means that new muscle is likely being created. Now, we don't have that drilled down yet, but that's sort of the initial read. New muscle is created, means the heart's squeezing better, means that potentially we're delaying some of the adverse effects of Duchenne on the heart. So stay tuned on that. I think that's going to be some very exciting data to follow up with. And I'm enjoying learning about heart disease and Duchenne patients along with you. So finally, and not unimportant at all, is the safety results. We've done a total of 56 infusions in HOPE-2 to date. We had 2 serious adverse events in the forms of immediate immune reactions. One of the patients developed an anaphylactic-like response. He recovered fine and went home the next day, probably telling his friends about it. There were no specific signals identified that were triggered in that patient. Another patient had a much milder event. It was already after we had initiated this pre-medication regimen, and it was a very mild event. We observed him overnight because, patients should be watched if they've had any type of an allergic reaction, and he was absolutely fine. Did not develop anaphylaxis, which means that the pre-med regimen appears to be working. And when we did some analysis on this patient, we found that he was a very hypersensitive kid, responded to almost everything that we exposed him to. So we're fairly convinced that the pre-med regimen is working well. Very stable, standard pre-med regimen. Yeah.
So, Linda, a little bit about anaphylaxis here, that these patients had an allergic reaction, perhaps not to the cells themselves, but can you kind of go into that a little bit and talk about what is the regimen you use to pre-treat these patients so it doesn't happen?
Absolutely. Thank you, Pat. So it turns out, I'm going to start with the first question. It turns out that it's very common when foreign proteins are delivered, and that can be if you're getting an antibody therapy or anything else. Some people, and we don't know who you are, but some people will mount this very strong initial immune response. It is not likely in this situation to be to the cells themselves because it happens so rapidly. In some of the patients, it happened within 2 milliliters, tiny, like literally like a drop hitting their blood. So not enough time for them to mount a response to the cells. This again is something that's considered to be very common. And so because delivering biologic agents are very common now, every hospital has what they call a pre-med regimen for this. And we selected the one that's used at UC Davis. Craig McDonald, our national PI, was familiar with it, and it's extremely simple. Basically means the day before, the patient takes a high-dose steroid pill. He takes some steroids, I think, that night and the next morning, and then when he comes into the center, he gets intravenous steroids some more and some antihistamines, standard stuff like Benadryl, and this absolutely attenuates that response. So it's a very easy thing to do. It's very common, not unexpected, and all the immunologists that we have consulted with have said this is a very standard practice and should not impede the path of this therapeutic to commercialization or being used in patients. Since the initiation of the regimen, we've done 30 infusions, and the only event that we had was the one that I described where the patient had a very mild response but was a very hyperallergic patient or young man, and so was very lucky that he got that because it showed that it was able to attenuate that response. Does anybody have any questions, Pat, or do you, before I move on, off of pre-med?
Yeah, no. So just a couple questions for us to clarify. This is a mutation-agnostic study, so mutation did not enter into the equation in terms of the inclusion criteria. But I think maybe it would be helpful to say, are all of these individuals in the study on the same or a dose of steroid as their stable, steady state?
Yeah, are all the patients getting the same dose of steroid? They get weighted dose of steroid.
Same dose regimen or different doses in regimen?
Different dosing regimens. So some of the patients, and I don't have that data in front of me, but I can dig it out. Some of the patients, there's a couple of centers now that are doing this weekend on, weekend off, weekday off kind of regimen with steroids. And I think some of the patients might have been on that. I actually don't know that answer right now. I do know they were all on stable doses, and the only changes that we made that we didn't make, that we were allowed, were weight-based. So if a kid came in and he needed an adjustment in his steroids because of weight, that was allowed. Otherwise, no other changes were allowed. And I can get back and get the data to steroid regimen.
Yeah. No, I just wondered if there was a consistent regimen, and I'm really happy you talked about the fact that if there was a weight change that you were adjusting the dose prior to the study as well. So thank you. I think you can go on, and then we'll come back with questions at the end.
Of course. So finally, this is, the colloquial take-home pitch or take-home slide. What we saw is really overwhelmingly positive. The most gratifying aspect of the study has been that we've seen corroboration of the data across different muscle groups. So we've seen skeletal muscle improvement, pulmonary improvements, which suggest diaphragmatic improvements, and improvements in cardiac function through systolic wall thickening. Just to recapitulate, because I've given you a lot of data in a very short amount of time, we saw statistical significance in the performance of the upper limb 2.0 at 6 months, P.0389, and a strong signal at 3 months of P equals 0.0591, so nearing significance but not quite there. We look forward to seeing data from 9 and 12 months, which we don't have enough of to analyze yet. We also saw improvement in grip strength at 6 months and strong trend in tip-to-tip pinch strength at 6 months. So we're encouraged that there seems to be multiple measures of skeletal muscle performance improvements. We saw statistical significance of inspiratory flow reserve at 3 months. And trends in peak expiratory flow, percent predicted, a measure specifically called out by the FDA as an indicator of diaphragmatic strength improvement in our FDA meeting minutes. So we're very gratified to see those trends and look forward to seeing them continue. Those trends were at 6 months primarily. We saw a positive trend in cardiac measures. Cardiac structure and function were only measured at 6 and 12 months. We don't have enough data 12 months to analyze, so the data that you saw was only 6 months data. We don't want to put kids in an MRI too often, so we just do 2 time points plus the heart change is slower than other muscles that are impacted by the disease. So the conclusion, and the most important conclusion, is the interim data seen in HOPE-2 is consistent with the FDA's guidance on requirements for potential registration, PUL plus other measures of skeletal muscle improvement, and supportive pulmonary or cardiac measures. We are going to get in front of the FDA as soon as possible. I had to get through this week. We just finished literally today our DSMB meeting where we needed to make sure that they were comfortable with the data, and they are. So we are cleared to proceed, which we're very excited about, and we'll hopefully be treating the 20 patients that are already enrolled in the study very soon. As soon as you're able to get your visit scheduled, and my clinical team will be reaching out to the centers and the centers to their families very soon. At this point, I'll take questions and answers. I will tell you before we move on that until we talk to the FDA, we don't know much about registration. So I can tell you that this data does not appear to be enough for registration. The big issue is that it's 6 months data, not even just a small sample size. You guys are used to small sample sizes. It's more that we need to have 12 months data. So we're going to go forward as fast as we can to get the 12 months data from the 20 patients we already have. During that time, we'll also go meet with the FDA, and then as soon as we have a plan in place for what's coming next, whether there'll be another trial, it's possible because we've unblinded that we'll have to do another trial with the performance of the upper limb 2.0 as the primary efficacy, and then the other things that you've seen in terms of skeletal, pulmonary, and cardiac built in behind those to support the findings. It's also possible, I'm not ruling out the possibility that FDA will let us finish out HOPE-2 and use that as the registration study. So all things are on the table at this time, and I wanted to take a moment before we jump into your questions, and I know you have a lot of them, so I wanted to give you plenty of time for them. I wanted to thank you. I wanted to thank this community. This community moves mountains for their sons and for themselves, and it is an honor and a pleasure to have the opportunity to work in this disease state and to see if I can potentially make a difference. When I became a scientist long ago, this was what I dreamed of, was to be able to develop therapeutics that could not just treat one person, but could treat many. So thank you for volunteering your sons, your time, your efforts. These trials are hard. On families, I know getting to centers and going through all of these different tests. But it's important to move the field forward, and let's keep trying to make a difference together. Thank you all. Thank you, Pat. Thank you, Kaylin. And questions, I'm ready.
So, Linda, thank you for this. We're certainly encouraged by these results that we see, and I think it is very important to say this is a small sample set. This is 6 month data, and clearly the FDA is going to be interested in 12 month data. And I'm guessing that with the 12 month data, if you continue to see statistically important progress, that you might be able to consider that as a registration trial. So one question has come in. Can you talk about who's currently being dosed, and is the dosing on hold, or are you continuing with all these patients going forward? So the patients that are currently in the study are going forward, and there's a second question to this. The placebo patients, will you then begin them on active treatment? And then what happens next? Are you planning to enroll more people in this study, or are you confining it to these 20?
Yeah, so thank you, and I knew these were going to be the questions. That would be mine if I was had a family member impacted. So question number one, we want to go back into patients as quickly as possible. There is an infusion hold on at this moment. We haven't lifted it yet. We needed a couple of things to happen. One, we needed to see this data. We've seen that. We needed the DSMB to tell us we were allowed to go forward, and I just did that literally 45 minutes ago. So we need to find out how fast before we can get back into patients, and that's why I'm saying my clinical team will start to reach out to the sites as soon as we can, which should be very soon, to get these 20 patients back in for their continued infusions. In terms of reopening the study for enrollment, we're not going to do that until we meet with the FDA, which will probably be early part of the fall. Let's hope September, October, depends on when they give us a meeting, and they give us guidance as to what they want us to do. It's possible, because we've unblinded this trial, and because the community knows that it appears to be working, and the investigators that know it appears to be working, even though that nobody knows the individual treatment assignments of the patients, this sometimes can cloud data, and FDA doesn't like it, and they tell you that you need to go back and do another study and close this one out. We're preparing for that. We're writing a clinical protocol right now, and we'll have that ready to go if that's what they mandate. If they say we can open enrollment and use this as a registration study and just add a few more patients, we'll be ready for that as well. So stay tuned as to exactly what's going to happen over the next couple of months. I'll keep you very well informed. In terms of if you're a placebo patient in the clinical trial right now, I believe in the protocol we've written in that there's an open label extension available for you. And I think that all patients have to reach 12 months before that can actually then be the patients can then be started on drug for the obvious reasons of blinding and unblinding. But again, stay tuned on that because that might change if we can't use this data set for registration. So all things, this is early. I'll just say as a postscript to that question, we decided to do this webinar early without having all the answers because I wanted to give you the data. So unfortunately, I have good news and not a lot of answers to follow-up questions, but we'll keep you informed.
Yeah, thanks, Linda. So as you've shown in your data, and maybe we can go back to the slides where you have the placebo patients, and I'm trying to think, it was slide 15 or even before that. Can we go back there?
Yep, all right.
Are we going there?
It should be moving. I'm moving my slides. I see them.
Yeah, you're moving. You're doing well. You're doing well. Yeah, I think I think we're
just tell me where to start.
Yeah, okay. So let's see. Maybe go forward one. Let's go to 14. I think this is 13, go to 14. No, you went one too far. So wait, stop there. So here's my question about that. So these patients that have received the 2 doses, right, that we see at month 6, you have 6 patients who've received 2 doses of the CAP-1002, right?
Right.
And do you see the effect is cumulative or additive? So basically, are the cells from the first injection just, the exosomes degraded and gone, and then you give another dose to sort of bolster that? Or do you see a cumulative effect? Is it your belief that there's a cumulative effect here?
Yeah, so I do think that's the case. It's early days and early to say, but because we saw more significance at 6 months than at 3 months in almost all of the measures that we looked at, it seems that there is a cumulative impact. So I'm hoping we'll see that continue in the 9 and 12 months. This slide's a good one to stop on because you can see we only have 2 treated patients at 9 months, and one of those patients actually made it to 12 months. And so we don't really have a good answer yet as to what's happening at those time points. We're looking at individual patient data right now. And what I can tell you is that both of those patients sort of are following their own natural history. The 9-month patients and the one that gets to 12 months doesn't seem to be changing very much yet. So we'll see what happens with others.
Okay, and then you said that right now you're not dosing because you spoke this morning, to the Data Safety Monitoring Board, or the DSMB, which are usually a group of experts in the field, key opinion leaders, who you discuss what your findings are, and then they discuss whether or not you should go on. So my question is, so these patients that we're going through in month 6 that had 2 doses, now month 9 you have only 2, in month 12. So are you is there going to be any delay in those people getting on their scheduled dosing regimen as described by the protocol?
We are trying very hard to make sure that doesn't happen. It's one of the reasons why I'm, sort of rushing to release the data and do the webinar. And we had our DSMB meeting. We had it all teed up because we have a bolus of patients coming along for assessments at 9 and 12 months now. And so we wanted not to have delays. So my goal and what we're working very hard on, I have a great clinical team to try and get the patients in. If you're a patient in the trial, it'll be very important for you to get in as fast as possible for your scheduled visit so that we can get the answer here.
Great, thank you. And looking far ahead then, should this become a registration trial and in our hopeful event that this will become an approved therapy, we would see this as a lifelong therapy, that you would receive this infusion of the CAP-1002 every 3 months at that dose, 150, for the rest of your life. Is that what we're thinking about?
That's what I'm thinking about. There's always the possibility that we could potentially back off. Maybe it's every 3 months for a year or 2, and then every 6 months after that. We'll have to do phase 4 studies to figure that out. But for right now, the data that we have right now, we're operating under every 3 months, for the rest of your life. You come in, you get your 30-minute infusion, and you go home.
Right. Okay, and I know this is also a very small sample set, but were you, in looking at the 6 patients who received CAP-1002, did you see any individual variation in this? Did you see someone who did incredibly well, or a responder and a non-responder in that very small sample set?
No, so one of the things the statistician explained to me when I first got the data, and I should share with you as well, is that one of the reasons why we see statistical significance with such a small sample size is because of how small the variability is. So the reason clinical trials are usually bigger is because you're trying to damp down variability. You're trying to make that less of an issue, and the more patients that you have, the less variability you typically have impacting your data. So the fact that with this very small sample size that we're seeing statistical significance suggests that there is a small amount of variation and therefore a very tight data set.
Okay, great, thank you. And if we go back to do one of the pulmonary outcomes, the pulmonary function, the IFR, can we go to that slide?
On my way.
There you go.
Here.
Okay, so it seemed like the placebo group improved at month 6. Is that what you're seeing as well?
I'm sorry, I didn't hear that last part.
At month 6, Linda, it looked like the placebo group and the treated group came up to about the same level, or am I misreading?
Yeah, so the means are looking about the same, but the variability is slightly less. We're trying to understand this data now. This data is really hot off the press, and as you know, Pat, better than anybody, some of the pulmonary function test data is very hard to analyze, so that's why we have Hank Meyer now doing the analysis of the raw spirometry data. What we saw, which we thought was interesting, is that we had, any improvement at all at 3 months, and then, similar mean changes at 6. What we're trying to understand now is, are those mean changes going to be persistent, or is the variability that you see in the placebo at 6 months driving the reduction in impact there?
Yeah, I don't disagree that the pulmonary function data is sometimes very difficult to really think through and how it all interacts. How the forced vital capacity and the IFR and the NIS and MEP and all that kind of wraps together. For those of you on the phone, Hank Meyer is at Children's Hospital of Philadelphia, and he's one of the experts in the field of pulmonology and Duchenne. We also have a question that said you said that the ITT, the intent to treat results, were almost exactly that per protocol. Was the ITT patient population also statistically significant, like the per protocol patients?
Yeah, I think there was. I have to go back. I'm actually pulling up the raw data as we speak. For which measure, Pat, are you asking for? The performance of the upper limb?
Yeah.
The performance of the upper limb. 2.0. There. In the ITT population, no, it was not statistically significant. It was 0.14 at 3 months and 0.13 at 6 months, so not quite statistically significant.
Okay, great. That's very, very helpful. And there is another question. As there is a wide standard deviation range from the data shown, if looking at individual patient data, is there a distinction for some patients? And we talked about this, responders and non-responders, and you just talked about the consistency of data throughout all those patients, right? So I think that we're looking at the fact that you have a group of patients in both the placebo and the active arm that were pretty standardized in terms of testing and results. Is that true?
Yes, that's exactly true.
Okay, so just looking forward, Lyndon, I know you can't say these, we can't say definitively. I think as a community, we're always going to say these things to you, and you're very used to them coming from us as well. As we see the data come forward, and as we see the 12-month data, and you have interactions with the RMAT folks at CBER, I think it's really important for you to be thinking of treating, the adult patients. Because in general, what we see is patients over the age of 18, other than Santhera, who's doing a wonderful job with Raxone or Idebenone. We really need to think about adults getting access to therapies, and so I don't know if they'll require a small study in adults or whatever, but we want obviously the broadest label possible as you go forward. So we want that on your radar screen, and I know that's always on it, but I have to mention it because I think many people on the line have are adults or have adult sons that they certainly would like to have the opportunity to take advantage of a therapy as this goes forward, should it continue to be show us positive results. And then I had just a question. How are you thinking? How young would you treat? Is that on your radar, or have you thought about that?
Yeah, so all of it's on the radar. It's funny when you were talking about the adults that and I'm sure the next question is going to be about the young ones. So, yes, I mean, I think ultimately our idea is to expand across the life course once we demonstrate efficacy. We ended up in a really sweet spot, and this is, serendipity favoring the prepared mind, because we have a patient population that is actually in a part of the natural history, unfortunately, where there's significant declines over 1 year. And so the important thing for clinical trials, as you all know, is to demonstrate a difference between natural history and what your therapeutic or your drug is doing. And so we have stuck with this patient population, one, because we really believe that they're the most needy ones, these people that are no longer ambulant and sort of have really nothing left for them and may have enough fibrosis that even gene therapy may not be as effective. But what we'd like to do over time, of course, is expand older into the adults. I mean, in HOPE-1, the average age was 17 or 18 years old, so, we've definitely seen efficacy in the older. And then, of course, eventually go backwards to the young ones. We're hoping that we can use this in conjunction with the gene therapy. We have done preclinical work with one of the gene therapies that are in the clinic now. I can't say which one because we're under confidentiality with them. But when we treat mice with our cells and with the gene therapies, it does not block the activity of the gene therapy, and our cells are allowed to work independently. So the cells work and the genes work, but they don't cross over and negatively impact each other. So you should theoretically, in your lifetime, be able to get the gene therapy and our cells for the fibrosis and the inflammation.
Great, thank you. And because there are antifibrotic drugs coming forward, there are certainly other anti-inflammatories coming forward. And do you see this working with all of those different opportunities that are looking at downstream inflammation, fibrosis, et cetera? And do you see this as a sort of a true combination approach to therapy as we go forward?
Absolutely. So, I think we all know that a complicated pathology as is created by Duchenne is not going to be something that is attacked by one therapeutic versus another. It's going to be a polypharmacy approach over time. And so our general concept is that we want something that will work in concert with the other therapies that are under investigation. It's hard to do those combined clinical trials. We did some of the gene therapy work with mice because we knew that was coming along, and I wanted to make sure that a cell and a gene could go into a body and not impact each other. But any of the other small molecules, absolutely.
Good, thank you. We have a question here that I'm just going to clarify about the pediatric disease, the PRV or pediatric research voucher that is awarded to companies in the rare disease space with an approval. So it's awarded as the approval happens. Capricor has not received a voucher at this moment, but would be eligible for a voucher should they be approved. So that's just a clarification question. And then we have a question coming in. Do you think the functional changes in the pool 2.0 are clinically meaningful, and how does that relate to patients and families? And I'll let you take it, and then I'll add to it.
Yep. So in terms of the first question, which I actually now have lost because I was thinking of the functional changes. So the first one was, remind me again, just give me 2 words.
The first one I clarified was the pediatric voucher that's awarded upon approval in a rare disease space. And the second question is, how does the pool relate to functional, to meaningful benefit, right? What do patients see in terms of the improvement that you've demonstrated? How does that relate to your arm function in terms of activities?
Right. So this is one of the most important questions, and it's one that's front and center in the minds of the FDA as well as, of course, patients and families. And the answer is relatively not straightforward, which is we don't really know. But what we're gunning for is about a 1-point change. And the performance of the upper limb was developed by a group in the United Kingdom called the ATOM group, and one of the leaders of that is Michelle Eagle. And when I showed this data to her, we now have a change of 0.6 at 6 months, so almost a 1 point or slightly less than a 1-point change. If we can get it to 1 point, it'll be considered even by the FDA as clinically meaningful. If we stay where we are but sustain it to 12 months, even that should potentially be clinically meaningful, because remember, what we're trying to do is slow down or stop the progression of the disease. We don't expect it to be turned around completely, but what we'd like to do is slow it down, and in other words, any improvement in function is a good thing. So, Pat, you can take it from there if you want. I think you have a little bit more background on that than me.
Yeah, so I can just talk to you about talk to the group about how the PUL, the performance of the upper limb, was developed. This was actually developed by World Duchenne Organization, UPPMD. It was called United Parent Project Muscular Dystrophy before it now evolved into World Duchenne Organization. More specifically, Elizabeth Vroom in the Netherlands and others, Michelle Eagle as mentioned, got together families with physical therapists and discussed how can we best measure the outcomes of the upper limb. And they started with looking at this about young men and talking about what's meaningful benefit to them and understanding what they felt was valuable, and then being able to divide the arm function up into the shoulder girdle, the elbow function, and the wrist hand. So this was developed by families at first and really evolved and validated with, obviously, the physical therapy community. But this was why it was so important, so that in testing and clinical protocols you can involve young men who have lost the ability to walk, and hopefully someday women as well, as we see X-linked inactivation on female carriers. So I think we have to understand that the PUL was developed as a means by families with experts really to look at the upper arm function in order to enable measurement in studies that would involve boys who had stopped walking. So, I think using the PUL 2.0 was recommended by the CBER, or Center for Biologics, who is responsible for cell and gene therapies, is a very useful tool, and certainly it provides meaningful benefit when we see improvements in the tool. So thank you for that, Linda. I think that's terrific. And one clarification on the pediatric voucher, it has to be a pediatric study, so the PRV, or pediatric research voucher, is really for approvals in the pediatric space, and you are looking at a pediatric population here. I did ask the question about going to adults, which would mean that if approved, you get a broad label and adults could be treated. And then I did ask about a sub-study. So I think, Linda, we have had the hour now, and I really do appreciate you coming forward and talking about this. Clearly, it's a small study. We need 12-month data, but the trends look like they're there, and we're really, really proud of that and really proud of the population you're studying. So thank you for that. And we'll look forward to another update soon after you speak with the FDA and decide what happens next. We'll look forward to learning about that and wish you well along the way. So thank you for taking time. I know your life is really hectic these days, and we really appreciate your time and your opportunity to discuss these interim results. So thank you very much. This has been terrific. And if other questions come in, we will pass them off to you, and we will get back in writing and then post on our website. So thank you once again. And to all of you who joined us, thanks for spending an hour with us. We know your time is precious, and we really appreciate it. Thank you again.